CAMK2 can phosphorylate its neighbor in this. And then like there are more mechanisms to that. But what we are interested is the mechanism called activation triggered subunit exchange. And for the activation triggered subunit exchange is this. I mentioned there are like 6 different states. This DPP state, the kinase domain of the CAMK2 subunit will dock on top of the association domain. Then this vertical dimer from this 2-hexameric ring will leave the holoenzyme and then diffuse through the cytoplasm, and it will find another holoenzyme, and then it will make it a tetradecamer. And main mechanism. And the reason that is important is it's active, and then if it is active, it can start phosphorylating its neighbors. So it could provide a mechanism to spread the activity of the CAMK2. Suggested way, the suggested way is the following: like a tetradecamer, it opens, it releases its active vertical dimer, and then it binds to an open dodecamer, and then like it becomes a vertical dimer. That's the whole picture. So we would like to see, like, can we make a model of this? And then can we estimate the subunit exchange parameters? Because I mean, like, we are putting new interactions on this CAMK2 model, and then we would like to see how quantitatively we can find those values. And there are 2 sets of experimental data that we find from literature, and we would like to make our fitting based on. And the first part of the first experiment is from 1998, and basically these experiments are for determining like how frequency of the input changes the CAMK2 activity. And there are many sets of experiments, but in general those experiments are taking place, let's say, less than 100 seconds. None of them are in long time scales. But if we come to the more recent experiments, the subunit exchange experiments, basically they have 2 different colored CAMK2 subunits. Some of them are red colored, they have this fluorescent, and then they have green colored, it's a different fluorescent. And then they immobilize them by using this PEG and then the streptavidin connection. And they start feeding the system calcium and calmodulin. And after a while, they just like take some sample, put under the turf microscope, and then they check like is there any colocalization of green and red? Because that means if there's a colocalization, that means in one way this immobilized subunits just holoenzyme and then like go through another holoenzyme and then like make the subunit exchange. So and then like there are some experimental data about it, but it's in general like 50-55. Like it goes up to 1 hour. So it's a time scale of data. So before we just jumped into any parameter estimation method, I just want to see that our model connections are good enough. So I made a parameter scan. I just like made a parameter scan with the parameter of this vertical dimer binding, unbinding, affinity. And then from that one, what I see that for short time scale data, it doesn't matter. Regardless, it will fit. Because like that's the experimental data that we have, and then like we have the same thing. But for the subunit exchange data, the bottom parts are our simulations, and the top part is the experimental data. We are far away from the reality. And then like I was wondering why this is happening, and then the reason is that it's because of this term. The phosphorylated calmodulin dissociation rate from CAMK2 is very low. The people model this because, like, I mean, when they're looking for the 6 seconds stimulation, they want a way, like there's a one-way transport. It starts from DUU, they gave to calmodulin calcium, and then it will get phosphorylated, and it will stuck at the state for 6 seconds, 100 seconds, and then like when it's stuck at that state, it will always active, and the activity level is always going to as in this S-shaped curve. For the case of subunit exchange mechanism, it does not work. So we think that we may also estimate this parameter. And well, we also need to train for the convolutional dissociation. And then we used rule-based modeling for this because pretty much there is no other way to do that because the state space is so large. If you try to generate this reaction network, it will, I don't know, I mean, there's like a paper about it, it's like 10 to the 44 different states, something. And then for the simulation of that sort of system, I mean, normally conventional network generation is not going to work for it, and then we are using this network-free simulations. And for the parameter estimation, a conventional method like Markov chain Monte Carlo, let's say, is not going to work because it needs at least 1 million of function evaluations. And each time that you evaluate this model with a good parameter set, it takes like 50 minutes. So if you make it, it's going up to like millions of CPU hours, and no one does not have it. And finally, the genetic algorithms is more scalable, and then like we can use this heuristic method to get some information about the parameters. And then BioNetFit is a tool published online available for the training of the rule-based models. And well, we tried, it did not converge. Because like basically this is like a chi-square this is a chi-square error function, and then like it starts from a high value, and then like we got here, and it did not work. And we tried to come up with some smarter way of solving this issue because it's because of the trials. Because like you need to go maybe more because like the energy landscape. I mean, well, it may be an energy landscape problem, but it's not enough because we only go through 40 generations. We can go more, and go more. We need to find out a smarter way of doing that. And then in 2016, I mean, like Dr. Bing Liu's work about the statistical model checking involves in a method called online model checking. So basically, in his models, there's a model checker, and he defines some logics in the middle of the trajectory, and then the simulation is getting stopped, and then like he checks that the logic is okay or not, and then if everything is off, just terminates that trajectory, and then like he's not spending anything extra on that trajectory. Because that's the biggest drawback of using the genetic algorithm, because at the beginning you're just far from your original or real parameter, and then like you're making a lot of model estimations, and then you're always simulating full trajectory. And for that purpose, I just want to identify this online model checking genetic algorithm method. And then for this one, basically you run a trajectory, you define a logic, and my logic is basically if it is the average experimental data, if my error is larger than 1 standard deviation of that particular time point, I'm just terminating that one. Let me start. To test this, I mean, like, I just don't want to jump into the ChemK2 directly. I would like to test this one in a simple model and hope that it works. And then this trivalent ligand and bivalent receptor model is one of the pathological examples of this network-free models, because like the ligand has like 3 binding states and receptor has 2 binding states, and then like this ligand can bind to the receptor, and then like because of the cross-link, it can go up to like an infinite size. And when I do this, I just like generated some synthetic data from an old model, from the model of the TLBR, and then our results looking quite promising actually, because for example, in a regular genetic algorithm, your objective function starts from somewhere high, it goes slowly down, and then like it will jump down to a very low level, and then like it will just go to a certain value it's converged. Our method, because in the beginning we are just going with a very elite population, because like we are just throwing many of the bad trajectories, and then like we are getting a very, very low objective function in the beginning, so that like we are not spending too many time on the bad trajectories. And if you look at this, the number of function evaluations that we make, I mean, this is regular genetic algorithms. Every step, it just calculates full trajectories. But in our method, in the beginning, in the first trajectory, you know, we are going up maybe 10 times more than what genetic algorithm does. But eventually, we just converge to the same point, and then like maybe like at 11 generation or something, we get a good result. And when we try to implement the same strategy for the ChemK2, we had some issues about that, like we are not able to stop and resume network-free simulations if you're a species more than 20. And well, it's a huge technical limitation, and that's a lot of work to fix it. So converge. But I mean, we have a good idea to how to fix that. And in summary, I mean, well, we started this work to learn something about ChemK2 because like it's a central molecule. But like we hit a bottleneck on the parameter estimation, and then like we come up with a good strategy to solve this issue. But I mean, in the end, we were not able to converge on the parameter estimation, but that's like how we ended up this work. And basically, in conclusion, for the dopamine transporter, heterogeneity in the surface distribution and the realistic geometry of dopamine neuron has a major effect on the dopamine clearance. And then, like the concentration of cocaine is not a big is a factor. And then, like for the amphetamine, the concentration is not a huge factor, especially at the concentrations that we are dealing with. The hybrid genetic algorithm online model checking methods can speed up simulations up to an order of magnitude, so maybe we can use it for future. And throughout my studies, I also worked on something else, and these are like some publications that I made. And well, I would like to thank my advisors, Dr. Yvette Bahar and Dr. James Fader. Throughout the whole process, they're helpful. They just taught me a lot of different stuff. They taught me how to be a good scientist. And then I would like to thank my committee members, Dr. Alexander Sorkin, Dr. Terry Sinowski, and Dr. Andreas Fenning. And specifically, I would like to thank to Dr. Mary Chang for all the good discussions and the advice about the dopamine transporter work. I would like to thank Dr. Bing Liu for his help with all this model checking, amphetamine model, and then Dr. Ethan Block for all the imaging studies with the dopamine transporters and all his contribution. And Dr. Tom Bartol, he taught me a lot about how to use MCell and how to do that large-scale simulations, and Dr. James Krieger for his input to all my studies, and Dr. Jose Montepia for his help on NFSIM, BioNetGen, and all the other stuff. And I would like to thank all current and past members of the Bahar Lab, Fader Lab, and current colleagues, the molecular genomics and pathology lab, bioinformatics. And the funding is there. Thanks.
Nice talk. I think my question is more like ask your comments or suggestion what we can learn from your drug reviews, models, and what we can, improve the current models. Currently, I think the output or results are, to me, it's a little bit, yeah. Can you give us some comments?
Yeah, definitely. I mean, well, the psychostimulants and the drug abuse is a big problem, right? And then maybe like the cocaine has a simplified way to affect the dopamine signaling, right? So I'm not sure what we can do. We can try like different concentrations of cocaine, maybe. I mean, I don't know, because it's a very simple mechanism. It's the mechanism of action is simple. That's the problem. But for amphetamine, I mean, sky's the limit. We can do a lot of different things, like because right now we simplify the mechanism as simple as possible, because we just choose two main functions. One of them is the dopamine efflux, and the other one is the endocytosis. And we add a spatial way to control these two mechanisms. It's not perfect, but it's like a good way to start, I think, like to understand the effects of effects of the psychostimulants in a spatial context. And I mean, you can always add more players to the intracellular part, because I'm trying to avoid it, because if you try to track all the intracellular molecules, it's so hard to simulate that sort of systems. It's already a huge model, and then like if you increase the size more, it will just take more time, and then I'm not sure how much benefit you'll get from that. But of course, you can add, let's say, ChemK2, because we are more familiar with its kinetics. And then, I don't know, maybe we can add more molecules that does not have too high copy numbers. Like we can understand how they affect the dopaminergic system.
do you expect to be able to some novel findings or from this kind of simple simulation? If it's too simple, do you really think we should continue to pursue this type of simulation?
I mean, sometimes you need to start with like things, right? And like the cocaine has a simple mechanism of action. I mean, I can just like simplify it more, complete, get rid of the diffusion scheme, and then like depending on the concentration of the cocaine, I'm just gonna destroy that dopamine molecules. But you will never see that half IC50 value has an effect at this content, because you will just reduce the total number of dopamine transporters based on your number of cocaines. But you don't see what is the effect of the diffusion, because like the bottom line is we are trying to make an in silico microscope, right? We are trying to understand how these events are happening in a spatial context where you have a real cell and with the other molecules. And then you will start with a model of this in the beginning. Eventually, you will of course add more detail. And with the current computation technologies or something, like we are trying to, press the boundaries as much as possible. And then I mean, even if it is simple, you should start simple and then, like, what's that, the Occam razor? You should start with the simple thing and then like add more details on it. And that is exactly what we do, because like we tried the dopamine transporter model, the dopamine reuptake model with the spatial context for the first time. And then when we have this model, we are trying to extend it by adding these drugs or adding some other players. And then eventually we will learn more, and then it will be a bigger model. And then it will, of course, answer more questions. But right now, this is the answers that we could get. I mean, without space, it's already challenging. And adding diffusion to something that challenging will make it more challenging. So I'm not sure, is it a good idea to add that right now? Maybe in future, of course, after we learn more about the model, maybe we can come up with like some simplified scheme, and we can use the space with this. Because whenever you go into the spatial context, you try to make it simplified. Because if you try to make it both complex and spatial, you will end up with simulations that is running for years, and then you don't get any result.
Reason I'm asking is Apple is developing the applications to take and change the time. And because time table is a lot of considering the shape of.
So you said the shape of the molecule itself, or the shape of the cells? Oh, spine. Okay, you're talking about the LTP. Yeah. I mean, I think. Which. Yeah. They're working. I mean, basically, the mechanism is the CAMT2 is getting activated, and then it will interact with some cytoskeleton regulating molecules, and then the spine is getting bigger as a result of it. And then Nosky and Tom Bartol has some studies on it, like how they can use this activation to increase the size of the box. And it's a complete different problem in spatial simulation. It's called the dynamic geometry problem, because your geometry is changing with your dynamics. Then, to couple them is a challenging problem, and I mean Tom and Terry and Jim and people at the Dispersed Supercomputing Center has been working on this problem for a substantial amount of time, and it's not a simple task. Yeah. Yes.
Yeah, extracellular and non-extracellular synaptic space services a lot, because thinking, what about people say that dopamine can improve plasticity at glutamatergic synapses? Do you think there'd be enough to activate the high affinity receptors on those regions?
So let me reword the question and tell me if I understand correctly. So you want to activate glutamatergic synapses with dopamine? And then you're asking like, can you activate the high affinity receptors on the glutamatergic cell? Basically, you will have a cell which has both glutamate receptors and dopamine receptors, and then you release a dopamine molecule from somewhere distant, and then it will diffuse through that place, and then like it will activate the receptors, right? And it's possible, but the main thing is like for example, we built this three-dimensional reconstruction of the dopamine transporter, right? Because we got the data for that. But like to make some sort of thing like this, like some glutamate transporters, some dopamine transporters, and then like some of those cells, if there's some electron microscope images, maybe it's possible to do that because you need the geometry. Like you cannot come up with all right, I made up this geometry, and then this is probably a good representation of my system, and then I'm just going to simulate my system in the setting. No, it's not a great way to do this. Like if you have good data, you can just make it 3D and like add your reactions.
You can't make some approximation based on the data that you already have.
What's that?
We can't use the data that you already have from your simulations to say if you go a certain distance away from the dopamine release site, is there still enough dopamine around to potentially do it?
Oh yeah. Well, I mean, basically we track all of them. We just like count, like I have all these box, and then like I divide this box into 1,000 small boxes, and then I count the number of dopamine inside it. What is the volume of this small box, of the extracellular volume, and then like I made all my concentration calculations based on it. So MCell enables you to track that sort of things, and of course you can.
Yes, just one last follow-up. So you've got all these small boxes. Why not just put the receptors in there and see how much?
Actually, that's a great question, Jim. And I mean, I think I talked to Dr. Sorkin about it at some time, and then like we were just like talking about like having two-channel images of the same system, but like you need a model for that. I mean, for example, Dr. Sorkin has this hemagglutinin dopamine transporter mouse model, right? It's a state-of-art thing. It's almost like impossible to find. And like we are trying to come up with a strategy that we can tag two molecules in a living organism, which is quite challenging task.
No, you can do it. It's not challenging. It's not. I think you can do it. Why not? You just, you can uniformly distribute it in the first place.
Yeah, that's pretty much what we did. Yeah.
Exactly.
That's pretty much what we did, I mean. But yeah, it's not like explicit definition of the receptors yet.
Yeah, my point is just that it might be easier to actually just put the receptors in than to try to do a calculation that's.
Yeah,
sometimes the simulation can kind of answer questions for you that it's hard to get calculated in terms of some order parameter for the system is much harder than actually just some receptor aspect. That's just.
Yeah, that's a really good point,
Yeah.
but like it's always coming with the cost. You can add more receptors. You can uniformly distribute them. Like if you know like their surface density, you can add like 1 million receptors, and then your simulation time will just go down exponentially slower, and then you can get that. It's possible. Sometimes you need to choose your battles when you're doing that sort of things, right? Sometimes you simplify.
We just didn't think of it at the time, so then you have, and these simulations are expensive.
We didn't think about it. That original.
I add that actually, and then like I started to simulate, and then it's not.
That was the plan.
I mean, I try to come up with like a strategy to realistically put them on the brain, but like it did not work.
If there are no more questions from the audience, I think we'll do the public portion of this and have a committee meeting.